The gut microbiome, home to trillions of bacteria, viruses, and other microorganisms, has emerged as a powerful regulator of human health, influencing everything from metabolism and immune function to inflammation and even cognitive performance. With aging, the composition and activity of the gut microbiota typically shift, often in ways that promote frailty, inflammation, and disease. However, in centenarians and other exceptionally long-lived individuals, the microbiome appears to age differently. Rather than following the expected trajectory of microbial decline and imbalance, these individuals often retain a rich and diverse microbial ecosystem, marked by a high abundance of specific beneficial species such as Akkermansia mucinifera and Bifidobacterium. These unique microbial signatures may actively contribute to longevity by producing health-promoting metabolites, supporting gut barrier integrity, and fine-tuning immune responses. As people age, the diversity and stability of the gut microbiota tend to decrease. This reduction in microbial diversity is often accompanied by a rise in potentially pathogenic species and a decline in bacteria that produce short-chain fatty acids, SCFAs, key signaling molecules that regulate inflammation, metabolism, and epithelial health. In contrast, centenarians often exhibit a more balanced microbial profile, with surprisingly robust levels of SCFA-producing bacteria such as Faecalibacterium prosnitzii, Roseburia, and Butyrivibrio. These microbes generate butyrate and other SCFAs, which serve as energy sources for colonocytes, enhance the gut barrier, and exhibit anti-inflammatory properties. The sustained presence of these bacteria may help protect the aging gut from dysfunction and systemic inflammation. One of the standout microbial players in this context is Akkermansia mucinifera, a mucin-degrading bacterium found in the mucus layer of the gut. Far from being harmful, Akkermansia has gained attention for its role in maintaining intestinal barrier integrity and metabolic homeostasis. Its presence is associated with lower levels of inflammation, improved glucose regulation, and enhanced production of SCFAs and secondary bile acids, factors that collectively contribute to healthy aging. Studies have shown that Akkermansia abundance is often reduced in individuals with obesity, type 2 diabetes, and inflammatory bowel diseases, while remaining relatively high in metabolically healthy elderly and centenarians. This suggests that Akkermansia is not merely a bystander, but may actively support the longevity phenotype. Moreover, the gut microbiota communicates with the immune system in profound ways. Nearly 70% of the body's immune cells reside in or near the gut, constantly interacting with microbial metabolites and antigens. A healthy microbiome educates the immune system, promoting tolerance to harmless stimuli while enhancing defenses against pathogens. In centenarians, immune profiles often show a surprisingly preserved function, including effective T-cell and NK cell activity and lower levels of chronic inflammation, inflammaging. The gut microbiota likely contributes to this balance by regulating the production of cytokines and supporting regulatory immune pathways. Beyond SCFAs, gut microbes also produce a range of bioactive metabolites that influence systemic aging. These include tryptophan-derived compounds, neurotransmitter precursors, and secondary bile acids that modulate lipid metabolism and immune signaling. For example, microbial metabolism of dietary fiber and polyphenol leads to the production of urolithins, which have been shown to stimulate mitophagy and reduce inflammation in experimental models. In LLIs, higher levels of such metabolites may contribute to cellular health and tissue maintenance well into advanced age. Interestingly, the microbiomes of centenarians also reflect lifelong dietary patterns. In regions known for exceptional longevity, such as Sardinia, Okinawa, and Nicoya, diets are typically high in plant fibers, fermented foods, and minimally processed ingredients. These nutrient-dense, microbiota-accessible diets support a rich ecosystem of gut microbes, many of which depend on fiber fermentation and polyphenol metabolism for survival. Over time, such diets may promote a microbiome that not only supports digestion but also acts as a metabolic and immunological ally in the aging process. In addition to natural diets, certain probiotic and prebiotic interventions are being explored to replicate or restore aspects of the centenarian microbiome. Supplementation with Akkermansia mucinifera, Bifidobacterium longum, or butyrate-producing bacteria is being tested for its ability to improve insulin sensitivity, reduce gut permeability, and lower inflammation. Similarly, dietary fibers such as inulin and resistant starches can selectively feed beneficial microbes and promote SCFA production. While these strategies are still under investigation, they offer a potential means of supporting microbiome health and possibly biological age in the broader population. The microbiome of centenarians challenges the assumption that gut health inevitably deteriorates with age. Instead, it presents a model of microbial balance and resilience that may actively support whole-body health well into the 10th decade and beyond. These individuals remind us that the gut is not merely a digestive organ, but a dynamic, adaptable ecosystem at the center of the aging process. As research continues to map the links between gut microbes, immunity, metabolism, and aging, the prospect of microbiome-targeted therapies for longevity becomes more tangible. Whether through personalized nutrition, advanced probiotics, or lifestyle-driven microbial support, the goal is clear: to cultivate a gut environment that promotes not just longer life, but a longer life lived in health.